Hi everyone, this is the 7th lecture for your chapter Cell. In the last class, we discussed different transports across the cell, and lastly we finished with all the different functions of a cell membrane. Okay. In today's class, we will be starting with the next portion, which includes cytoplasm and a new term, which is endomembrane system. In the endomembrane system, we will be starting today with endoplasmic reticulum. So without much ado, let us start. Cytoplasm. To understand cytoplasm, there are few terms that are important. Okay. First term is sol. Now I know you have studied in chemistry what different sols are. Okay, what different colloids are. So sol is basically a colloid made with liquid and which has very small solid particles in it. Clear? So you know the meaning of sol now. From that term comes the word cytosol. Wherever the word starts with CYT, okay, CYT or CYTE, it means the cell. Okay, so cytosol means the colloid that you find in a cell. Fine. So it is made up of water, plus it has dissolved ions and sugars, sodium, chlorine, potassium, all of these will be dissolved. Okay, they are ions, they can dissolve very well in water. Then you have sugars, glucose, fructose, galactose, all of these sugars will also be dissolved in the water. Okay, so cytosol is basically water plus dissolved material plus small molecules like proteins which are insoluble. Okay, these are not soluble, so these can be considered as small solid particles. So such small solid particles like protein, tRNA, which is transfer RNA, nucleotides, vitamins, enzymes, etc. All of these are included in these small solid particles, and this is the reason why your cytoplasm is sometimes referred to as cytosol. Clear? Very good. Now let us see, what does cytoplasm mean? Cytoplasm is this sol, which is just the liquid portion and the small solids in it, the cytosol, plus cell organelles except nucleus. Okay, all the different organelles that you see here in a cell. Okay, it could be endoplasmic reticulum, it could be Golgi complex, it could be mitochondria, centrioles, ribosomes, all of these are included in cytoplasm except nucleus. Okay, except for this nucleus, you include everything. The water part, which is cytosol, plus the cell organelles will make cytoplasm. The next term, which is rarely used these days, but it used to be very famous when the science was new. Okay, so it is called protoplasm. In few old textbooks or old syllabus, you will still find this word called protoplasm. Protoplasm is basically cytoplasm plus nucleus. So the whole cell except the cell membrane, okay, everything enclosed by a cell membrane is called a protoplasm or protoplast. Clear? So these are the different terms. What we will be studying is actually protoplasm. Okay, we will even include nucleus. But before we do that, we'll leave nucleus for the end. Okay, it will be the last organelle we will study. So before that, we will be studying all the cytoplasmic components. Fine. I hope this much is clear. 90% of cytoplasmic matrix is made up of water. So 90% of cytoplasmic matrix is actually made up of water. The rest of it is obviously organelles and different solid particles. Fats, now some of them will dissolve, the other like tRNA, nucleotide, vitamin, enzymes, proteins, all of these are hydrophilic in some way or another. So they can be found in water. Okay, they can easily stay in water. But the one molecule, the one biomolecule which cannot gel well with water is actually fat, right? So fats occur as emulsion. What is an emulsion? This also you must have studied in chemistry, right? Emulsion is basically two different phases. Here the phases are water and oil. So these two phases are somehow with use of emulsifiers. In cell, emulsifiers can be phospholipids, they can be proteins. Okay, different emulsifiers are found in cell. So with help of such emulsifiers, the fat are dissolved, okay, or they are present, or else they are present in form of globules, right? Just think of adding a drop of oil in a glass of water. You can see how spherical form it takes. So that spherical form, a very small sphere of oil, can be called a globule. Okay, so the fats can be present inside a cell in form of small globules. Okay, and obviously fats are present as component of various biomembranes, which we just studied in the last class. Some functions of the cytoplasm. Before jumping to the functions, this diagram, or this, it is actually a painting. This painting I wanted to show you, because in a cell, when you think of cytoplasm, just because it is made up of water does not mean it is empty. Okay, if you look here, this is the plasma membrane. Here you can see the desmosomes. Okay, and this is a mitochondria. Here is your DNA, and this is the nuclear envelope. Fine. So between the plasma membrane and the nuclear envelope, whatever you see is actually plasma, okay, or your cytoplasm. So in this cytoplasm, can you see so many different things are present? There is cytoskeleton, there are different vesicles, there are different organelles, small molecules, large molecules, many proteins. So it is actually very crowded in the cytoplasm. Okay, so this is the image I want you to remember. Cytoplasm, just because it is made up of water or because it looks clear does not mean it is empty. Fine. Okay, so some functions of cytoplasm. It contains the raw material. Okay, the raw material which is required by different organelles to function well. Fine. Then it does the exchange process. Okay, the unconnected cell organelles, because let us consider here is a mitochondria and here is endoplasmic reticulum. For example, some protein from endoplasmic reticulum needs to reach mitochondria, but because they are not connected, what will happen? You need to transport it, right? So this transport, this exchange will take place through cytoplasm. Fine. Then products and waste produced by different organelles will also be added to this cytoplasm. Then biosynthesis. Biosynthesis is basically anabolic reactions. So a number of biochemicals like fats, remember this, fatty acid biosynthesis, then nucleotides, carbohydrates, proteins, coenzymes, and a lot of different things are synthesized in the cytoplasm. I think you already know that at least protein is also synthesized in cytoplasm, okay, on the free ribosomes. The protein gets synthesized. So the protein translation happens in cytoplasm. That is also a part of biosynthesis. Then you have different catabolic activities. This was anabolism, synthesis. Then you have catabolism. Catabolism is to break something, okay, break a larger molecule to get smaller molecules plus energy. So cellular respiration, like glycolysis, of course, when we say cytoplasm here, I'm talking about cytoplasm matrix. Okay. Otherwise, in cytoplasm, you have to include all the organelles also. But organelles we will be studying separately. So right now, whatever I'm talking about is the cytoplasmic matrix. Okay. The area which is outside the organelles, whatever the volume is filled outside the organelles, that is all cytoplasmic matrix. So in cytoplasmic matrix, you have cellular respiration occurring, which includes glycolysis, anaerobic respiration like fermentation. It could either be lactic acid fermentation or it can be alcohol fermentation, right? Then pentose phosphate pathway or triple P pathway or hexose monophosphate shunt. All three of these are the name of the same pathway. Okay, so that also occurs in the cytoplasmic matrix. See, here it is very important that you connect different chapters and different topics you have read about or understood or even you have already learnt about. Okay, when you connect few chapters, different topics, that is how you build your base. Okay, that is how you will become stronger. So always whenever I'm teaching, I will try to mention, okay, which all chapters you will find a description about some of these. So for example, cellular respiration, this will be your chapter number 14, okay, in your 11th standard. So if you recall, try and recall glycolysis, anaerobic respiration, all of this actually happens in the cytoplasm. So this is how you correlate, okay? And yeah, so it is always better to learn like that. Fine. Now that you are preparing for NEET, this is how strong you have to be, okay? Then cytoplasmic streaming. Cytoplasmic streaming matlab, the cytoplasmic matrix is always in motion. Okay, though you cannot see in a lot of cells, we will see a video now in which you can actually visibly see that the cytoplasm or the cytoplasmic matrix is actually moving. Okay, this matrix keeps moving so that all the material inside gets distributed evenly. Okay, think of a cell. What if on one end you have glucose molecule and on the other hand you require it to go to, okay? For example, let us say on the other hand you have mitochondria. So the glucose molecule has to travel from one end to the other end. How will it travel? So this becomes easier when there is cytoplasmic streaming. Just let me show you, okay, how it is happening. See, these are the plant cells, and in these plant cells, because the chloroplast is green in color, you can actually see them moving. Okay, you can see the chloroplast moving. So this is how the cytoplasmic streaming works. Of course, this image has been sped up. Okay, so it is not this fast, but just for the sake of understanding, a lot of time has been compiled into the short section. Okay, so you can see the chloroplast moving around, and this is how all of the material gets distributed evenly throughout the cell. Okay, now if somebody asks you, or you are curious and you want to ask me that, madam, what is carrying these things around? Okay, how is this cytoplasmic streaming, how is this movement occurring? So I will show you another image. Think of this thing. Okay, there will be microtubules which act as highways. Then there are some proteins. This one, okay, this mister who is moving around, okay, 1 foot ahead, then second foot ahead, it is actually a protein called motor protein. Remember this protein, it will be important, okay, later on when we study. So these motor proteins can actually carry a whole vesicle, a whole organelle, and what they do is they just keep on moving around such highways. And that is how the cytoplasmic streaming happens, okay, with help of cytoskeleton, which we will study later on, and with help of such motor proteins. Of course, this does not happen without the use of ATP, so ATP gets hydrolyzed here with each step this protein takes. Another thing, cytoplasmic streaming, I teach in 2 other chapters. First chapter being transport in plants. Okay, when I discuss transport in plants, cytoplasmic streaming comes in discussion. Intracellular, okay, within a cell, the transport is happening because of cytoplasmic streaming, so I included there. Also, in chapter number 15, which is plant growth and development, I include cytoplasmic streaming there as well when we discuss different plant movements. Okay, so cytoplasmic streaming is also a type of plant movement that you see within a cell. Fine, so it is important to remember. Yeah, one more thing, cytoplasmic streaming is seen in hydrilla. Okay, hydrilla, which is an aquatic plant. I don't know if you have ever noticed or observed it under a microscope, but if you get a chance, please do. And cytoplasmic streaming is also called cyclosis. So with that, I will end this slide, okay, and we end the cytoplasm. I hope this much was clear. Now, in the cytoplasm, we are done with discussing about the cytoplasmic matrix. We will discuss about the organelles from now on. Okay. So many cell organelles are actually connected because of their functions, which are coordinated. Okay, their functions are coordinated. So it is just better for some organelles to be connected in one way or another. Such endomembrane system, endo means inside, membrane, okay, they are talking about cell membrane. So inside the cell membrane, endomembrane, there is a system, okay, there is some system, which now you will be able to understand why we call it endomembrane system. It includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So these four organelles are interconnected. Okay, they rely on each other for their functioning. Starting with the first endomembrane organelle, which is endoplasmic reticulum. Okay, now this name also we will break it down. Endo means inside the cell. Okay, inside. Plasmic, inside the plasma membrane. Reticulum, wherever you hear this word reticulum, it means network. Okay, it means net. So because this endoplasmic reticulum, the organelle looks like a network inside a cell, it is named such. Okay, endoplasmic reticulum. It is the largest membranous network inside a cell. Fine. One more thing. From reticulum, I realized you will study in chemical coordination, endocrine system. Whenever you study, kidney has adrenal cortex. Adrenal cortex has three layers, right? Zona glomerulosa, zona fasciculata, and the third layer is called zona reticularis. So that reticularis, again, reticularis means network. Okay, so remember this word. Sometimes what happens is just because you know the meaning of the term, okay, you can answer a lot of questions. Fine. Chalo. Let us begin with what endoplasmic reticulum is. It is not an organelle which you can see by light microscope. Okay. Chloroplast, yes, you can see by light microscope. Nucleus, yes, you can see. Cell wall, yes. But endoplasmic reticulum, no. If you try and look under a microscope, you will not be able to see the endoplasmic reticulum. Okay, so it can only be seen by an electron microscope. And here is the image you get from electron microscope. All of this network that you see is actually endoplasmic reticulum. Okay, you can see the cell almost looks like a fingerprint, right? So each of these things that you see, long tubes, they are nothing but endoplasmic reticulum. It is a 3D, okay, it is in 3 dimensions. 3D interconnected system of membrane lined channels. Okay, so these are like channels. Channels, I hope you know. Think of canals or tunnels. Okay, this is just another term for channel. Fine. So channel means some path, okay, some tunnel. So this tunnel is actually lined by membrane. Okay, so it is covered by membrane that run through the cytoplasm. At some point, it is connected to cell membrane and nuclear envelope. Okay, whatever the nuclear covering is there, the endoplasmic reticulum will be attached at some places. Okay, and it will also be attached to the cell membrane at some places. Then plasmodesmata, we studied desmotubule, right? Just recall the cell wall, and I think even in cell membrane we discussed when we were discussing the junctions, right? So plasmodesmata, it contains ER in form of desmotubules. From now on, whenever I mention ER, it means endoplasmic reticulum. Okay. It divides the intracellular space between into two compartments, okay, luminal and extraluminal. Now one more thing I want you to clarify is inter, INTER, and intra are different. Okay, intra means within and inter means between two things, okay, between two or more things. So you can say intercellular when there is something between two cells, and when you want to talk about something within the cell, you say intracellular. Okay, so it divides the intracellular space, which is the space inside the cell, into two parts. Luminal is whatever the lumen, okay, lumen means cavity. So endoplasmic reticulum has such cavities, okay, the canals. So that part is called luminal space, and whatever is there outside is called extraluminal, okay, the rest of the cytoplasm. It forms major part of endomembrane system and has 30 to 40 times more surface area compared to the cell membrane. If you consider this normal cell and you try and find out what is the periphery, okay, then you will find that compared to that area, okay, the area of a cell membrane, the inner area which is made because of endoplasmic reticulum, okay, the surface area made available because of endoplasmic reticulum is much higher. How much higher? 30 to 40%, okay, 30 to 40 times, actually 30 to 40 times the surface area within a cell is higher, okay. Endoplasmic reticulum is extensive. Extensive means elaborate, okay, very highly developed. So it is extensive in metabolically active cells. The cells which are metabolically active, you can find a lot of ER in those cells, like cells of pancreas. You know, pancreas is a mixed gland, right? It has endocrine function, which is it secretes insulin, glucagon, and somatostatin. And it also has exocrine function, which means it secretes all the digestive enzymes, right? So because these pancreas are very active, they have a lot of endoplasmic reticulum. Same is the case with liver. Liver also has a lot of functions. So it in the cells of liver, which are called hepatocytes, also have a lot of endoplasmic reticulum. It is simple in storage cells like adipose cells, okay. When you talk about cells which do the function of storage, okay, for example, adipose cells or adipocytes are the cells which store fat, okay. Here is the diagram, and this big bubble you see is actually a fat droplet, okay. And if you notice, the cytoplasm is very reduced, and in that cytoplasm also the endoplasmic reticulum is still further reduced, okay, compared to the diagram we saw in the previous slide. In this one, in this cell, you can see that it is very much reduced and very simple in such storage cells. It is absent in sperm cells, okay. In sperms, endoplasmic reticulum is absent. In egg, endoplasmic reticulum is absent. Mature RBC of humans, okay, and mammals, because we do not have nucleus, we also do not have endoplasmic reticulum, okay. So in RBCs, we do not have a lot of organelles. One of them is endoplasmic reticulum. And the cells which are at rest, okay, which do not do a lot of functioning, a lot of protein synthesis, such cells will have less or almost zero amount of endoplasmic reticulum. Now we talk about the endoplasmic reticulum structure. I had to give you some homework. Okay, wait. Yeah. So my question for you is, why in egg cell, ova, whatever you want to call it, why in egg cell endoplasmic reticulum is absent? In sperm you can understand because it is so small, you cannot fit endoplasmic reticulum, okay, it is very small in size, which we saw in when we discussed sizes and shapes of cells, right? But egg is the largest cell in the human body. Then why does egg not have endoplasmic reticulum? This is my question for you. You have to find it out. This is homework, okay? Polish your brain. Chalo. So endoplasmic reticulum structure. If this is the nucleus, nuclear membrane or nuclear envelope has two layers, okay? There is outer membrane here and there is inner membrane. Fine. So with the outer membrane, this endoplasmic reticulum is connected. Can you see here? Okay. So this is the nuclear membrane, and with this nuclear membrane, the endoplasmic reticulum is connected. Each such channel, okay, each such sac-like structure is called cistern. which is written here. Okay, cisternae. Fine. Each such structure is called a cisternae. It is lined by a single layer of membrane. Okay. This can be 40 to 50 nanometer in diameter. Okay, so that much space is found between the two, and that space is called lumen. Okay, whatever the white space you are seeing between the two layers is called lumen. Part of endoplasmic reticulum is also found in such globular forms, okay, such spherical form. These are also lined by a single layer, and they are called vesicles or microsomes. Okay, they are 25 to 500 nanometer in diameter, and they are just sacs, okay, sac-like structures, rounded spherical sac-like structures, and they are called vesicles or microsomes. Finally, you have a few which are actually very thin and somewhat branched, and these structures are called tubules because they actually look like tubes, right? They look like tubes, so they are called tubules. So remember these three terms. Endoplasmic reticulum is made up of cisternae, it is made up of vesicles and tubules. These three parts make an endoplasmic reticulum. Now there are two types of endoplasmic reticulum. Okay, one is called rough endoplasmic reticulum, RER. Why is it called rough? Because if you look at the surface, you will find that it is a little bit uneven. Okay, you'll find the surface to be uneven. This upper diagram is showing it in 2D and the lower diagram is showing it in 3D. Fine. So the upper surface, whatever the surface of endoplasmic reticulum is there, it is uneven. It looks granular. Granular, in Gujarati you call it danadar. Fine. So it looks that kind of granular. So it is also known as granular endoplasmic reticulum or rough endoplasmic reticulum. Now you may ask, what are these granules? So these are nothing but ribosomes. Then there are some types of endoplasmic reticulum which do not have ribosomes. Okay, so they are smooth. You can see how smooth they are. Okay, in both these diagrams. So they are called smooth endoplasmic reticulum or agranular endoplasmic reticulum. We start with the smooth endoplasmic reticulum, SER. It has smooth membranes without the ribosomes. You see here, this is an electron micrograph, okay? And here you can see very smooth lining of the cisternae, and these are nothing but smooth endoplasmic reticulums. Fine? They are found in cells which synthesize and store glycogen. Liver cells do the storage and synthesis of glycogen. So remember this, okay? Glycogen is nothing but it is a polysaccharide of glucose, okay? So glycogen is stored in liver cells. It is also synthesized in liver cells. So if somebody asks you, where exactly in a liver cell, then the answer will be SER. Second thing, fats are synthesized and stored in liver cells. Again, hepatocytes. See, I'm mentioning liver cells so that it is easy for you. But later on, I will keep on introducing the proper term, the scientific term for liver cells, which is hepatocytes. Okay. So in hepatocytes, some amount of fat is stored and a lot of fat is synthesized. Okay. So synthesis and storage of fat in liver cells is done by SER. Adipocytes, which again store a lot of lipids. They have smooth endoplasmic reticulum doing the function. Sterols, steroids, and steroid hormones. All of these are synthesized and stored by the smooth endoplasmic reticulum in hepatocytes and adrenal cortex. Adrenal glands, okay, which you find above your kidneys. They have two parts. The outer part is called cortex, the inner part is called medulla. The outer part is secreting different steroid hormones, okay, mineralocorticoids, glucocorticoids, sex corticoids. So all of these hormones, steroid hormones, are made by the smooth endoplasmic reticulum. Clear? In muscle cells, it stores calcium. Now this, when you study locomotion and movement, okay, that chapter, you will be studying muscle contraction. So in muscle contraction, you know that calcium plays a very important role. So this calcium, where is it coming from? Part of this calcium is coming from the endoplasmic reticulum, smooth endoplasmic reticulum, which has sort of differentiated, and it is now called sarcoplasmic reticulum. Okay, it does the function of storing the calcium inside a cell. Okay, the calcium is not moving around in the cytoplasm, it is stored inside this sarcoplasmic reticulum, which is nothing but a modification of smooth endoplasmic reticulum. It is mostly made up of vesicles and tubules. Remember, we studied three parts: cisternae, vesicles, and tubules, right? So these three parts. In that smooth endoplasmic reticulum, you will mostly find it is made up of vesicles and it is made up of tubules. Clear? All of this what you see are mostly tubules. New endoplasmic reticulum when developed from existing endoplasmic reticulum. Okay, if somebody asks you, okay, where does new smooth endoplasmic reticulum come from? How is it originating? Where is it originating from? So the answer is actually it originates from rough endoplasmic reticulum. The rough endoplasmic reticulum which sheds all the ribosome, it becomes smooth endoplasmic reticulum. Clear? It can also come from the plasma membrane or nuclear envelope. Okay. Now we move on to the rough endoplasmic reticulum. It has ribosomes attached to their outer surface. Okay, you can see here this is one part of the cisternae which has been shown. Okay, whatever the inside you see, that is the lumen of endoplasmic reticulum. This is the outer membrane of endoplasmic reticulum, and on this membrane you can see ribosomes are attached. Okay, these pink or purple colored structures are nothing but ribosomes. Because of the ribosomes, rough endoplasmic reticulum is engaged in synthesizing proteins and enzymes. Okay, it itself does not synthesize, but because it has a lot of ribosomes attached on the top, these ribosomes synthesize, okay, the proteins, and these proteins are packaged, and they are 3D structure, okay, is formed inside the lumen of endoplasmic reticulum. Fine. So you can see here the blue colored strand is nothing but your mRNA. Then they have small pores. Okay, there are pores made on the endoplasmic reticulum so that the ribosome, whatever protein it is synthesizing, can enter inside the lumen. Fine. This protein enters the lumen, it gets folded here, somewhat modified also, and then it gets packaged even in the endoplasmic reticulum. It is abundant in the cells which are actively synthesizing secretory proteins. Secretory proteins are the proteins that the cell secretes. Okay, have you heard of insulin? So insulin is secreted by the beta cells of the pancreas, right? So these cells can be called secretory. Then goblet cells of your digestive system are secreting, let us say, some mucus, okay, or other cells are secreting, let us say, pepsinogen. So secretory proteins, pepsinogen is an enzyme, so this enzyme is packaged, okay, and synthesized in endoplasmic reticulum. Fine. RER helps produce lysosomes with Golgi apparatus. Okay, it alone cannot do it, but whatever enzymes are present inside the lysosomes, okay, these are synthesized in the rough endoplasmic reticulum. It is mostly made up of cisternae. We saw that smooth endoplasmic reticulum is made up of vesicles and tubules, while the rough endoplasmic reticulum is made up of cisternae, and tubules are very few. These are the differences between SER and RER, okay, smooth endoplasmic reticulum and rough endoplasmic reticulum. This time I will not be discussing these differences, okay. I want you to do the homework, okay. Your homework will be from whatever I have taught, that plus whatever you read, you have to make such kind of a table, okay, a table of differences between smooth endoplasmic reticulum and rough endoplasmic reticulum. That is another homework for today. Right, now we move on to the function of endoplasmic reticulum. These are the total functions, okay, of smooth plus rough endoplasmic reticulum. So the first thing is it provides a large surface area inside the cell for various physiological activities. Okay, some things are not in your syllabus otherwise, um, but I would just like to mention, okay, that these endoplasmic reticulum membranes, they also have enzymes, okay, and these enzymes can catalyze a lot of reactions. Okay, so because of this larger surface area, various physiological activities, enzymatic activities also occur. Okay, then they provide mechanical support to the cell. Right, because there is a lot of endoplasmic reticulum. It will provide some sort of support to the cell structure and shape. It keeps the various organelles in their possession. Okay, because they are in sort of a network, they will keep specific organelles in their possession. Okay, then desmotubules, which are found in plasmodesmata, they control the movement of material between two adjacent protoplasts. Okay, between two adjacent cells. Signals, proteins, a lot of things are exchanged through these desmotubules. Then they conduct information. This part I will discuss again when we talk about Golgi complex. For now, just know that they conduct information. Okay, they will take information present outside the cell and conduct it towards inner side to nucleus, or from inside, whatever the signals are there, the information is there, can be taken outside. Okay, or also from one cell to another, from one part of a cell to another. Okay, so they conduct information. It provides membranes to nuclear envelope after telophase. So after telophase, obviously you require membrane to form around the DNA. So this membrane, okay, this nuclear envelope is actually synthesized or produced by endoplasmic reticulum. Okay, the endoplasmic reticulum membrane itself will be making the nuclear envelope. We shall see that in some parts or in some ways, even Golgi complex helps in the process. Then it gives membrane to Golgi apparatus for formation of vesicles and lysosomes. Okay, so Golgi apparatus also is supposedly it originated from the endoplasmic reticulum. Okay, we do not know few origins, okay, how these organelles are coming into existence, but we do know that somewhere or the another they are related to endoplasmic reticulum. So from endoplasmic reticulum itself, Golgi apparatus would come into existence along with their vesicles and lysosomes. It also gives rise to spherosomes. Spherosomes are nothing but they are small microbodies which store this one, lipids, okay, and vacuoles. These are all formed from endoplasmic reticulum. Complexes of proteins and lipid to form lipoproteins. Okay, have you heard of LDL, low density lipoprotein, high density lipoprotein? Um, HDL is the bad cholesterol, LDL is good cholesterol, something like this you might have heard. So these HDL, LDL, etc., these molecules are actually proteins plus lipids. Okay, so such complexes are called lipoproteins, and lipoproteins are made in endoplasmic reticulum. So here we end today's chapter. You have been given two homework. One is to find out why there is no endoplasmic reticulum in egg cell, and a second homework is to make that table, okay, of comparison between the smooth and the rough endoplasmic reticulum. Apart from that, I I have almost stopped discussing the questions at the end of the chapters, lectures actually, sorry, because you have been given the assignment, okay, which has a lot of questions. So it will be better if you yourself solve these questions. If there will be some special questions which I find interesting or important, I will mention them in the lectures. Fine. So that is all for today. See you tomorrow.